Okay, so chapter 5, we are now working on energy and enzymes, specifically enzymes in this chapter. So if we look at what we've already covered, we've already talked about fireflies and how the energy for the cell to do work. We have talked about energy is the ability to do work. There's two types of energy, kinetic and potential. We've talked about the laws of thermodynamics. We've talked about how chemical reactions can either be endergonic or exergonic. And we've talked about how cells carry out thousands or millions of reactions. It's called their metabolism. Energy coupling is the process by which exergonic reactions provide the energy for endergonic reactions. We've talked about how ATP is the energy molecule that cells use to do work. So we're trying to generate ATP or we're using ATP to do a reaction. What I want to focus on is how enzymes function. Enzymes are what speed up a cell's chemical reaction by lowering the energy barrier or the amount of energy needed for the actual reaction to occur. So if we were to look here at this diagram, you can see very clearly that here's your reactants, here's your product. So these guys are trying to jump over this. What is this that they're jumping over? This is an EA barrier. It is the energy barrier that acts like a giant wall that prevents the reaction from just naturally occurring. So the reactants have to climb over this energy barrier before they can reach their products. What an enzyme does is an enzyme pushes down the wall, and it makes it easier for the reactants to become the products. Because of that, when we talk about enzymes, we're talking about proteins. Enzymes are proteins, and their job is to act as a catalyst. And what I mean by saying they're a catalyst is they decrease the energy of activation needed to begin a reaction. So here is a graph format of the same diagram we just saw. So here's your reactants in an uncatalyzed reaction. They have to go all the way up this hill before they can become the products. Whereas if you see the red line, if you're an enzyme, notice that the amount of energy we needed to kind of get over the hill and allow the reaction to naturally occur is much lower. And because of that, they lower the amount of activation energy, and that's why we call them a catalyst. Now, there are specific enzymes that catalyze each cellular reaction. So enzymes have a very, very specific shape, as all proteins do. However, there is one shape or one enzyme that does each function. And so the shape of an enzyme determines what chemical reaction can occur inside of it. When we talk about enzymes, we have new vocabulary that you guys have to become familiar with. So, for example, if we talk about reactants in a reaction, we now call them substrates. If we talk about the chemical reaction, the location in which it occurs becomes the active site. And so because enzymes are very specific for what they do, we consider it to be like a lock and a key. There's only one key that fits in every lock. Therefore, there's only one substrate that can fit to the active site of an enzyme, which then allows only one chemical reaction to occur. So if we were to look here at this diagram, you would see that there is steps going on in an enzyme reaction. So here is our enzyme, and it has an empty active site. So this is the active site. And if we look up here, here's our substrate. So step kind of 1 or 2 in the reaction is you have an empty enzyme with an empty active site. You allow the substrate, which is sucrose. Remember, ose is a sugar. So we're going to bring in our substrate, and it fits perfectly into the active site. That's the lock and key, also called the induced fit model. And so now we have them together. Because the enzyme is holding the substrate there, it becomes easier for water to come in and hydrolyze or break the bond, which is the next step in a chemical enzyme reaction. So here's your enzyme. Here's our substrate. We're using water to hydrolyze or break the bond. Now that we've done the chemical reaction, our products over here are going to leave the active site, and we're going to be left with an enzyme that has become unchanged by the reaction. It hasn't been affected or altered at all, and its active site is now empty, and it's free to do the entire process again. So again, an enzyme is a catalyst that lowers the amount of energy needed for the reaction to occur, and it does so because it provides a binding site for which the enzyme reaction can happen. Now, there are things that will affect an enzyme, and that becomes very important as we kind of move forward. Temperature is a major one. So if you look here at this top graph, the optimal temperature for human enzymes is 38 degrees Celsius, or about 96.8 degrees or 98.6 degrees Fahrenheit. Whereas if we were to look at the same thing, these are high temperature bacteria that are found in like hot springs. And so if you notice, the optimal temperature for these bacteria is roughly 78 degrees Celsius. These bacteria or enzymes would not function well in our bodies, and our body enzymes would definitely not function well in a hot spring. So temperature has a major effect on how enzymes do their job. Another factor is pH. pH can also affect how enzymes work. If you look here at a pH of 2, which is the pH of your stomach, the enzyme pepsin, which is found in your stomach, hence the word Pepto-Bismol, pepsin, operates optimally at a pH of 2, which is good because it's found in our stomach. Whereas trypsin, which is another enzyme found in our body, it operates about a pH of 8, which is optimal for it. That pH is a standard pH outside of our body or inside of our body. Another factor can be cofactors. Cofactors can be inhibitors, or they can be coenzyme inhibitors. And these are things like vitamins, minerals, the trace elements that we kind of say are important for your body, and then we don't kind of expand on that. These guys bind to an enzyme, and when they bind to the enzyme, they either help the enzyme function or they prevent it from functioning, therefore altering the enzyme activity or the ability of the enzyme to do its job. To kind of talk more about this inhibition, there's two types of inhibitors. There can be competitive inhibitors or non-competitive inhibitors. Competitive inhibitors, what they do is they actually take the place of the substrate. So what they'll do is, here's our enzyme, here's our normal substrate. Notice the substrate fits perfectly into the active site right here. Well, if you were to look at a competitive inhibitor, here's our competitive inhibitor, it's binded to the active site, which then prevents our normal substrate from binding. If it can't bind, the reaction won't happen, and then we won't produce our products. So competitive inhibition literally takes the place in the active site. Whereas non-competitive inhibition or inhibitors, what they do is they bind to a different part of the enzyme called the allosteric site. When they bind, it changes the shape of the active site, which then prevents our substrate from binding. It no longer fits. We've changed the lock, so the key no longer will go in. Those are some types of inhibitors that can happen for enzymes. The big thing is to make the connection, because a lot of poisons or pesticides or drugs like Tylenol or Advil that we use, they work on enzymes. They work to prevent an enzyme from doing their job. We can kill plants, not humans, by stopping enzymes involved in photosynthesis, and that's how we have many of our plant poisons such as Roundup.